Good morning, everybody. Sorry we can't be face-to-face, but in the age of COVID, we're taking advantage of distance learning technology to present some of the research going on in my laboratory. My name is Dr. Robert Allen. I'm a professor of forensic science in the School of Forensic Sciences at Oklahoma State University Center for Health Sciences in Tulsa, Oklahoma. I am by trade a forensic geneticist, and what I'm going to talk to you about today is some of the work that we have been doing that is relevant to the concept of time. So let me just get things going here. So the investigation of a crime by a forensic scientist usually involves providing answers to four basic questions that allow for the reconstruction of a crime and the events that transpired leading to a crime. And ultimately, the four questions are who, what, where, and when. And if you can answer these four questions, you can often identify the perpetrator of a crime and subject them to prosecution, thereby maintaining public safety. So the question of who, at least in 2020, is an answer that is readily obtained through the analysis of DNA, looking at variability in our DNA and thus tracing the source of biological samples associated with a crime back to the person who participated or perhaps was responsible for the crime. Question of what, in terms of a forensic scientist world, is usually involved with identifying tissues or body fluid stains that are recovered as evidence from a crime scene. And we now have technologies that are much like the home pregnancy test technology, immunodiffusion chromatography, with the use of monoclonal antibodies that allow you to identify semen, blood, saliva, even urine, sweat, all based upon protein components that are present in those body fluids that will react with highly specific antibodies. We also can identify quite often a particular body fluid through microscopy, understanding the morphology of cell types that compose a particular tissue that may be present in that evidentiary sample. And the newest approach on the block these days is messenger RNA profiling, exploiting the tissue-specific collection of messenger RNAs that are present in cells comprising a tissue or a body fluid. And those patterns of gene expression can be exploited to identify that particular tissue type. The question of where is quite often self-evident just by looking at the crime scene. Of all these questions, the question of when is the one for which we have the fewest tools, and yet questions of when are often quite important in understanding the events that led to a crime. And so that's really where the research I'm going to talk about today originates. Back in 2014, a PhD student in my laboratory at the time, Kate Weinbrecht, and I decided that it would be interesting to see if we could estimate the age of biological material recovered as evidence from a crime scene. And our approach to beginning this project involved the use of next generation sequencing of all the RNA transcripts in blood, semen, saliva, and vaginal secretions that had been stored for varying lengths of time in the laboratory. And the question was, can we follow the decomposition or the degradation of RNAs, messenger RNAs, in these body fluid stains, dried body fluid stains, as they are stored over long periods of time, up to a year in this case, in the laboratory. So through the use of RNA sequencing, we could look at all the RNAs that were present, and based upon the sequence data which is an indirect reflection of the abundance of particular transcripts in these stains, we could ask whether or not RNAs disappeared from the transcriptome in a predictable fashion, and if so, do they disappear in a way that could be used sort of as a biological clock? And what we found was, yes, indeed, RNAs do disappear from the transcriptome of dried body fluid stains. And so we wanted to devise, using this RNA sequencing data, quantitative polymerase chain reaction assays, which are much cheaper, much more straightforward to perform in a forensic laboratory. And interestingly enough, in looking at the RNA sequencing data, we discovered that the sequencing read depth, which is an indirect measure of abundance, that the read depth of the 5' end of the messenger RNA disappeared at a faster rate than the read depth at the 3' end of that message. So, and data for this is shown here in this slide down in the bottom left, and you can see these histograms, the height of which reflect how many times each of the nucleotides in that region, in this case, beta-actin messenger RNA, how many times each of those nucleotides is actually sequenced in that reaction. So the higher the peaks in the histogram, the deeper, the larger the number of reads. And you can see at time 0, you have at about the same peak heights, if you will, from the 5' end of the message, which is on the right side of the rightmost series of peaks in the histogram, versus the 3' end, which is the leftmost. And after 180 days, clearly the 5' end has decreased. So this led to the design of a Qpcr assay, where we actually studied the abundance of the ends of single transcripts rather than quantify a transcript that changes in abundance against one that is presumably stable, a housekeeping gene or something, which is sort of the normal way people quantify the abundance of transcripts or any sort of DNA, any nucleic acid that is changing with time. So this led to the development of an assay, a Qpcr assay, in which we quantified the 5' end of a messenger RNA transcript, say the first 70 to 90 base pairs from the 5' end, versus another transcript of 75 to 90 bases from the 3' end of the transcript. The notion being that as it degrades, these fragments would remain inside these dried stains and would continue to decrease in abundance as the blood stain began to decompose. So as these curves, as you can see in these curves, this is 4 different transcripts present in blood stains: LGAL-S2, CLC, S100A12 down in the lower left, and beta-2 microglobulin. If you're at all familiar with RNA quantitation, beta-2 microglobulin is a message that is present in virtually all tissues. It produces a protein that associates with histocompatibility antigens and other membrane components, much like associates with histocompatibility antigens, at least much like immunoglobulin light chains associate with immunoglobulin heavy chains in antibody molecules. So as these curves increase going from left to right, the abundance of 5' end of these transcripts is disappearing from the transcriptome. And as the curve goes up and then levels off, the abundance of 5' end of the transcripts is below the limit of detection for our quantitative PCR platform. So this work was published by Dr. Jun Fu and myself in 2019, and it allowed us a very sensitive and reliable, we felt, approach to quantifying the abundance of RNAs in dried evidentiary type samples and blood stains, certainly in this case. And so to test this hypothesis, we created some stains. So we knew what their age was. We gave these stains to a technologist and said, as unknowns, and then using these curves for, in particular, S100A12, for example, we can plot the delta CQ value of our unknown on this curve and extrapolate, make an estimate of sample age. So when we did that, here's results for 8 stains, and we've got the actual age on the left, and then going to the right, the estimates made with the LGAL transcript, the CLC transcript, the S100 transcript, and beta-2 microglobulin. B2M, of course, is only good for very short periods. If you recall, the curve, it degrades very quickly from the 5' end and then levels out after about 6 to 8 weeks of incubation. And these stains, by the way, were stored in the laboratory in the dark under controlled conditions. But the point I want to make here, especially with the S100A12 transcript, is you get very good estimates of sample age based upon a lot of data that I won't actually talk about today, but statistical analysis of differences in delta CQ values for stains of differing age. We ultimately concluded from this work that the window of accuracy for any estimates for samples up to a year of storage was about 2 to 4 weeks for the first 6 months and about 4 to 6 weeks for 6 months to 1 year, probably even 6 months to 18 months or 2 years. Again, using the S100 transcript, we felt we had had made a great deal of progress in being able to estimate the age of at least blood stains recovered from crime scenes. So as I said, all of the storage here in the study I've talked about today this far is stains, blood stains, dry blood stains stored at room temperature in a controlled environment. However, that's not really very real world. So the next phase of the project was designed to try and evaluate the effects of environmental conditions on the rate of RNA degradation in blood stains, because evidence sometimes may be found in the trunk of a car parked in the hot sun, or may be found in a very cold environment, and all our environments in between. So we wanted to look at the effect of degradation. First challenge, though, was to create a stable environment in which we could control relative humidity and temperature. So the humidity part was initially kind of a challenge until I stumbled onto a publication from the early 1970s in the microbiology literature, where investigators were trying to assess the effects of relative humidity on the growth of bacteria. And in this one paper I discovered, the take-home lesson from that publication, at least for me, was that the discovery that if you have a sealed environmental chamber containing a beaker with a solution of a saturated salt, you can create a relative humidity defined percentage. So, for example, if you have a sealed chamber with saturated sodium chloride at equilibrium, you will get a relative humidity of 75%, really not terribly influenced by temperature. Magnesium chloride about 35%, and lithium chloride about 10% relative humidity. So here are pictures of 3 of our humidity chambers. You can see they contain blood stains that have been dried and deposited on blood stain cards. And then there's a big card that we found from Scientific Supply House that sort of gives you a ballpark range of what the relative humidity is inside these chambers. Sort of like pH paper, and you can see the one on the far left has a lot of pink in that line, indicating that the humidity is high, about 70 to 80%. The one in the middle is the magnesium chloride. The pink only goes up about halfway. And the one on the far right is lithium chloride. which shows virtually no pink at all. And these, the increments on that scale are increments of 10% relative humidity. Once we get these jars set up, we can then put them in 37-degree incubators or leave them at room temperature or put them in a 4-degree refrigerator. So ultimately, we ended up with 9 different environmental storage conditions. So the experiment was as follows. We prepared blood stains on collection cards. And then a group of these stains was stored in one of 9 different environments, 3 relative humidities and 3 temperatures. At selected times, RNA is extracted from duplicate stains initially, and then at 1, 2, 4, 8, 16, or 24 weeks of storage in that condition. When a time point is reached, the chamber would have to be opened, blood stain card taken out, quickly excise the stain, replace the card with the remaining stains back in the chamber, reseal it, and then put it back in its storage temperature. So there was a brief interruption of the environment within the chambers, but it was probably reestablished within 4 to 6 hours. So at each of the time points, we'd extract total RNA, reverse transcribe it into complementary DNA using oligo-DT and random primers, and then quantify the abundance of the 5' and 3' ends of these 3 transcripts using our Qpcr assay. And the question was, can we follow or can we notice any effect of temperature and our relative humidity on RNA degradation rate? So here are 3 of our storage conditions in terms of temperature: 37 degrees, 20 degrees, and 4 degrees. And then the colored curves in each of these panels represent the relative humidity. And I've got them listed here in trying to consider this data and digest this data. One wants to try and have a description of an environmental condition that's defined by a single mathematical value. So in thinking about this problem, what came to mind was the use of a heat index value. And a heat index value, if you watch your weather, especially in the summertime, weathermen will often give you a combined value, a value that combines both temperature and relative humidity into a single number. And so we're exploring that right now as our approach to combine both humidity and temperature into a single entity. Anyway, take-home lesson from this study is that both heat and humidity contribute to an accelerated rate of RNA degradation. The most severe and the most rapid rate of RNA degradation occurs at 37 degrees and 75, excuse me a minute. Oh, that's gonna sneeze. 37 degrees and 75% relative humidity. You can see that the rate of degradation of the 5' end of the S100A12 message increases very, very quickly and actually levels off after only about 4 weeks of storage. Whereas at 20 degrees and 35% humidity, the conditions sort of reflecting what we had in the laboratory, the curve is still increasing out to 6 months of storage, pretty much like we saw previously in our studies with blood stains initially. So the interesting thing here is that at 4 degrees, degradation almost halts completely. You can see a little bit of increase in the delta CQ value at 4 degrees and 75% relative humidity. But at 4 degrees and 10% relative humidity, things are virtually stable over an entire 6-month storage period. So for any of you in the listening world that are studying RNA in dried evidentiary type samples, the take-home lesson is that once you collect your sample from a scene, stored at 4 degrees in a dry environment to preserve the integrity of those transcripts for long periods. So how do we factor all of this environmental effect into an algorithm that will allow us to continue to estimate the age of the stain? Well, we we haven't gotten there yet, but we've chosen to start off by comparing degradation rate constants. And a degradation rate constant in this discussion is basically how long it's the maximum delta CQ value that is achieved during storage. All right, so when the curve gets to its highest value, the delta CQ value there minus the delta CQ value at time 0 divided by the amount of storage time it takes to get to that maximal value. And so if we compare these rate constants, if we control for temperature and do comparisons of rate constants for different humidities, or control for relative humidity and assess the effects of different temperatures, the take-home lesson from all of this work is that if you compare at a single temperature, 75% humidity versus 35% humidity, you get about a 2 to 3-fold increase in the degradation rate at the higher value. Likewise, if you have a single humidity and compare the effect of temperature, again, it's about a 2 to 3-fold increase in the rate of degradation of these transcripts at a higher temperature. So we're still dealing with evaluating this data to figure a way to incorporate it into, as I said, some sort of algorithm that retains the accuracy in estimating the age of a stain. Clearly, you can get the environmental conditions from the National Weather Organization. The cycling up and down of temperature and up and down of humidity is another variable that we're going to have to try and incorporate into our algorithm. But that's where we're going in the future. Now, another interesting fact is that once degradation starts, if you shift the sample from a high temperature, high humidity, sort of the blue line here in this curve, for the S100A12 transcript again, if you incubate it at 37 degrees and 75% humidity, and then at some point shift that environment to 4 degrees in a dry environment, you can shut down degradation. So it is reversible in the sense that you can promote degradation or retard or stop degradation altogether just by, and it happens fairly quickly once you make that temperature shift to the more stable environment, degradation halts, which to me suggests that it may be a chemical process is involved here in degradation as opposed to enzymatic. So, temperature and environment. Another part of the environment is the substrate that a body fluid stain resides on or is deposited on. So we wanted to assess the effect of the substrate as well. And substrates can include clothing or surfaces, metal, ceramic, wood, plastic, even carpet. And so we created blood stains on all of these different substrates and then stored them for periods of up to 6 months in the laboratory, again, under dark conditions. The results of that study, I'm just going to show you a couple of surfaces, which include carpet, and denim. And the take-home lesson from all of our substrate studies is that there really is no effect on the rate of RNA degradation, depending upon the surface when compared against blood stains created with stain cards that we're using sort of as our control. So the effect of the substrate is not very significant. It really doesn't have much of an effect on the rate of RNA degradation, which is good, because that would be, if it did have an effect, that would be a further complicator or distractor in trying to develop an algorithm with which to estimate time. Well, the newest thing the newest question we've asked is, if we can estimate what day a stain was created on, can we estimate the time of day a stain was created? And we thought that the way perhaps to investigate this question was to exploit rhythmic changes in gene expression that are associated with the circadian rhythm. And there's a tremendous amount of literature out there describing genes whose expression seems to vary over the daily cycle. And so our approach was to create blood stains and to assess the abundance of messenger RNAs transcribed from these genes over the 24-hour daily cycle. So the experiment was, somehow we managed to recruit a large number of volunteers who were willing to prick their finger, create a blood stain on a collection card every 4 hours around the clock. And we had, I want to say, 20 to 30 males of all different ages and about the same number of females who were participating, who were willing to volunteer for this study. So once we create a stain at a select time during the day, that stain within 1 to 2 days would be transferred back to the laboratory, RNA extracted from it, cDNA prepared, and then quantification of the individual transcripts originating from genes associated with the circadian rhythm would be quantified using Qpcr. In this particular set of experiments, we developed Qpcr primers that targeted the middle of each of these transcripts, because from all of our previous research, we knew that that was the most stable part of a messenger RNA, was the least likely to be degraded during that 2 hours of storage, or the time between the stain was created and the time that the RNA was extracted. So we got all this data, we analyzed it by sex, whether it's male or female, also by age in 10-year increments from 20 to 60 years of age. Interestingly, here's some of the data that was produced, and we had most of the transcripts that we looked at yielded results that either were unchanging over a 24-hour period or produced data very much akin to what you see in the top right panel there for the per3 transcript in males. Huge error bars. Now, it's important for you to know that these error bars do not reflect technical variability in our Qpcr assay. Rather, these error bars reflect biological variability among the 20-odd males that were tested. So it's kind of interesting that there's such widely varying levels of gene expression for genes that have been suggested to be associated with the circadian rhythm, that in males for this particular gene, the rhythm is all over the place. However, if you look at that same transcript in females, you see fairly tight variability in the time points. And the data for CQ values from noon to the evening on the right side of that curve are significantly different from the CQ values on the left side of that panel. So it would be possible, using this marker in a female blood stain, to distinguish whether it was if that blood stain was created in the afternoon or in the evening or early morning. Likewise, another transcript, the TPOH, which is tyrosine metabolizing enzyme that ultimately ends up involved in the production of melatonin, which of course is typically high in the evenings when you get sleepy. Again, those are statistically significant differences in CQ values from the early evening, 10, 11 o'clock, versus the early morning. So we're not where we need to be yet. We're still looking at additional transcripts that seem to show cyclic variation in abundance inside blood stains. But we feel that at least proof of concept here, if we have a whole lot of these markers that we can analyze in tests, that we may be able to discriminate when during the day within a couple of hours a stain was created. So that's where we are right now. I want to acknowledge the people in my laboratory that have contributed to the studies that I talked about today, and to emphasize once again that time is an important concept to the forensic scientist. And ultimately, we hope that from a single body fluid stain recovered from a crime scene, we can determine whose it is, we can determine what it is, whose it is, and when it was deposited. And all of those questions are going to be very relevant to our investigation of the crime. So thanks for your attention. I will take any questions, I guess, by email after those of you in the audience think about what I've said today, and I'll do my best to try and answer them. Thank you very much.